Okay, good morning, everybody. Why don't we get started? First, I just want to clarify that from last Wednesday's lecture, I didn't finish all the slides that I put in that slide presentation, if you were watching, but only what I covered. I covered up to Parkinson's disease, and we ended with that video of Michael J. Fox. That will be the only thing covered on the exam. So don't worry about Huntington's disease. Don't worry about the videos associated with, there was a deep brain stimulation video that you might want to look at just because it's very interesting, but that won't be on the exam, okay? Today, what we're going to do is I'm going to do a little review of the motor system in general, both motor system and motor diseases, and then we'll start on the learning and memory part of the lecture. That will be on the exam, but I'll be very specific about the aspects that I want you to be familiar with the exam. I did not post questions because I wasn't sure how far we were going to get, but I will post questions to give you and guidelines for what you need to know about this. I'm not going to make you memorize all the intricacies of learning and memory. It's going to be just the basics, and we'll mainly jump into that next Wednesday. Oh, you remember this coming Wednesday we have an exam, of course, but next Monday there's no class. There's no class because there's a big Society for Neuroscience meeting that I have to go to, and so we all get a little break for Monday. So the next lecture after Wednesday's test is the following Wednesday, so that's on your syllabus. Let's see, same format. The exam will have the same format as last exam. It's going to be true, false, fill in the blank, multiple choice, and then a series of short answer questions. It's going to be more challenging, this exam, than the first exam. Why? Because there's more bigger concepts. There's fewer lectures that we're covering on this exam than the last exam. But there are more complex topics that we cover, the similarities and differences between the sensory systems, the contrast between the sensory and motor systems. So that is, those are the announcements for today. Oh, and then the last announcement is, just so you know, that clock is wrong. It's about 15 minutes late, so don't use that clock as a gauge for the progression of the class. So our topic for today is kind of starts out a new direction in our lectures, and the third set of lectures that we're going to be covering today, and that is cognition, learning, and memory. But before I jump into these lectures, I thought I would do a little review for you, particularly for the motor systems and motor diseases. So your TAs tell me that you guys spent a lot of time going over all the sensory systems. How are you guys feeling about sensory systems? Okay? How many people feel really good about sensory systems? Kind of good. Kind of good. Okay, there's a few kind of good. The rest of you are feeling scared? Scared about sensory systems? Okay. Okay, okay, fear can be a good motivator. That's good. So let's start with those sensory systems. Let me see if I can give you some guidelines to add to, I know, the very nice guidelines that the TAs have been giving you in their review sessions and your conversations with them. Also, let me emphasize that the TAs are available for your questions. I am available between now and Monday at 11 o'clock when our lecture is. So feel free to come and talk to us. That's the best way to learn this stuff and get a feeling for whether you understand this stuff at the level that we want you to understand it. Okay, so a kind of a organizing format that I used through the sensory systems are these 5 key principles of the sensory systems, where transduction is defined as the conversion of outside energy into changes in membrane potentials. Those largely result in changes in action potentials So how outside energy is translated into action potentials. Okay, so we talked about that in the somatosensory system. What is the transduction mechanism in somatosensation? Anybody? How is touch translated into action potentials? Yes. Very good. Mechanically gated ion channels. So that is the answer. So you need to know what that transduction mechanism is for all the different senses. Does everybody know where to find that information? Put your hand up if you know where to find that information about transduction. Everybody? Okay, those of you that didn't put up your hand, why don't you know where to find that information? Or you're just not putting up your hand? Any thoughts? What is confusing you about transduction? Questions about transduction? Any questions? Okay, so let's continue with just the review then. So we talked about somatosensory transduction. We spent a lot of time on visual transduction. Everybody needs to know that the cells that transduce in the visual system are? Sorry, I didn't understand. Rods and cones. Ganglion cells help transmit the information because the rods and cones are the ones that are taking that light and translating it into changes in membrane potential, okay? So ganglion cells are good. That's part of the visual pathway, but it's not the transduction. It's not the cells that do transduction. Remember the definition of transduction. It's the conversion of outside energy into changes in membrane potential. So what cells are getting the outside energy and translating it? It's really those most outermost cells. The outermost cells in the visual system are rods and cones. And the outermost cells in the somatosensory system are all those different kinds of receptors that then use ion-gated touch-sensitive ion channels to change touch into action potentials. Question?
So just to specify, the photoreceptors of bipolar cells fire graded potentials, while the ganglion cells are the ones that actually fire the action potentials.
That's correct. Yes. Any other questions? Okay, so yes. How does a bipolar cell fire a graded potential? It is, it's not translated into an action potential. So graded potentials are simply changes in membrane potential that are going depolarized or hyperpolarized. So graded just means, just like the dendrites of a neuron from our first sections, they are giving EPSPs or IPSPs. Those are graded ion potentials. And then it gets translated into an action potential at the axon. But the bipolar cells are just giving graded action potentials. Yes, question in the back.
In the lecture, it said there's like a 1-to-1 ratio between a photoreceptor and a bipolar cell.
Mm-hmm.
But then it also has diagrams where there's like off-center and on-center bipolar cells.
Yes. So each photoreceptor is associated with a particular ganglion cell. And that ganglion cell can be on-center or off-center. But ganglion cells can also receive other lateral inputs. Okay? So it's the lateral inputs, not from the projections from the photoreceptors to the ganglion cells, from the photoreceptors to the bipolar cells that are giving those lateral interactions. We talked about amacrine cells and horizontal cells. Did Mike cover amacrine cells and horizontal cells in the lecture? Okay, sorry. But there are other horizontal interactions in, that's helping those more lateral interactions to cause the center-surround organization. So your first statement was absolutely correct, and the other interactions happen through other lateral connections within the retina, from the bipolar cells to the, from the rods and cones to the bipolar cells and the ganglion cells. Okay? Yes? acuity. Yes, yes. So that's very, very important in the foveal reason. That's correct. Yes?
Sorry, the previous question before that, is that what Professor Hawken meant by information in the retina travels parallel and serially? I didn't really get what he meant by that.
Yeah, so it travels in parallel because, so the example for why visual information travels in parallel is because there's different pathways that send different kinds of information through the retinal system. The M pathway and the P pathway are sensitive to different things. So in parallel, two kinds of information are coming through two different kinds of pathways, okay? But it's also serial because within that pathway you can have elaboration of higher and higher kinds of visual information happening at each step in the pathway. So that's what he meant by there's both a parallel pathway of visual information as well as a serial pathway. It's kind of taking advantage of all the different ways you can elaborate the light information getting into the visual system. Does that make sense? Okay, good. Any other questions? Yes?
Sorry, one more time.
Of course.
Horizontal transmission of information by the endocrine and horizontal cells. Endocrine and horizontal are both horizontal?
Yeah, they're going laterally.
Okay,
Yeah.
and they're going between which cells?
They are within the bipolar cell layer mainly. But we're not going to ask you a question about that because Professor Hawken didn't cover that. But that is how, it's important to realize that we're giving you a simplified view, and the way you fully understand sensor surround is through these other lateral interactions, even though we're not going to ask you about that. Okay? Transduction. So we talked about, you really need to know the 3 cells that do transduction in the somatosensory system, in the visual system, and in the auditory system. So what are the cells that transduce in the auditory system? Hair cells, inner hair cells. Both right. Okay. So what about outer hair cells? What do they do? Yes? Perfect. Perfect. Okay. And everybody gets that the mechanically gated ion channels in the somatosensory system, the rods and cones in the visual system, and the hair cells in the auditory system. Those are the cells that are transducing energy from the outside into action potentials. That is the definition of transduction. And they're doing it in similar ways, but we also talked about differences that they do it. So there was something unique about what those rods and cones, sorry, go ahead. Yes.
So the inner hair cells, those are the ones that are 90 to 95% of afferent input.
That's right.
Less actually than outer hair cells.
Yeah. So the inner hair cells are the ones that provide 90 to 95% of all the information that gets translated through auditory nerve 8. Okay, so that is basically the hearing information going through the inner hair cells. The outer hair cells provide the last 5, to maximum 10%. But what they're,
There are more outer hair cells.
there are many more outer hair cells, yes. So that's why you don't want to damage those inner hair cells. They're really fine and delicate, and there's not that many of them. And they're the ones that are helping you hear everything that you need to hear. Yes. Yes, you.
At some point go over the organization, spatial organization of the inner ear again, because I'm confused about the basilar membrane and where that is, and like where the hair cells protrude from one layer to the next.
Sure. Let's do that. I can just go and find. Let's see, 12, audition. Okay. So here, very quickly, because we have a lot to go over, I do want to get to the motor stuff. here's the cochlea. It looks like a shell, and there are 3 different tubes in this shell. How come this looks so bad? Oh, I know. Ah, okay, that looks better. There is the vestibular canal, the scala media or middle canal, and the scala tympani. And the key area that you need to know about is this middle canal, the scala media, because that's where all the important stuff lives. Okay, so let's look at what that important stuff is. The important stuff is in the scala media is called the organ of Corti, and it's made up of the hair cells, the supporting cells, and the basilar membrane. So here is a blow up of the scala media, just that middle part, and here is the organ of Corti that has inner hair cells that are towards the middle of the cochlea, the outer hair cells here, and then all these purple ones that are non-differentiated, those are the supporting cells. And the basilar membrane is on the bottom here. The basilar membrane is the one that's vibrating at different frequencies, where we said that, where did I put that? That the base of the basilar membrane, the part closest to the middle ear, is most sensitive to high frequencies. And this apex, or the very top, this is the rolled out cochlea. Did you guys, if you really want to understand this or really have a nice way, look at the animation that I put on the website for the auditory system. I didn't go over it in class, but it shows really cool, in a cool way, how this cochlea that looks like a shell unrolls and it ends up looking like this. And this is the base. That's sensitive to high frequencies, so it's going to vibrate with high frequencies. And you go all the way out to the top, or the end, or the apex. And this is sensitive to very low frequencies. Okay? So that basilar membrane is moving. And then we go back to our scala media and our organ of Corti. And you see what happens when the basilar membrane moves. You have these hair cells here. Now you know that the hair cells have these hairs sticking into another membrane, special membrane called the tectorial membrane. And what happens is when the basilar membrane moves, the hairs on the hair cells get pushed across the tectorial membrane and they get stimulated. And that's how the action potential happens. Okay? So that is the mechanism. Yes? It's not pushing. The tectorial membrane stays put. It's the basilar membrane that's moving, that's sensitive to different frequencies. Exactly, yeah. So it's like this, and my hair cells are going like this against the tectorial membrane. And that's what makes the hair cells move. Okay? Is that clear? Okay, so that is auditory stuff. Let's go back to, let's see. Here we are. Okay. Right. So we talked about transduction. You do need to know all of the neural pathways. So you were mentioning the ganglion cells. You need to know where they are in the whole visual pathway. Let's say up to the level of the thalamus. So if I asked you to write out all the steps of how light gets to the thalamus, what would you say? Light comes in, it stimulates what? The cells that transduce it. And then they project to what layer of the retina? Bipolar cells. And then they go where? Ganglion cells. And then they go where? LGN. That's the thalamus. There you go. That's your pathway for vision. Okay? Same thing for the somatosensory modalities. So there, let me just jump ahead. You remember what this is? Dorsal root ganglion. What kinds of cells live in there? What are their morphological characteristics? What do they look like? Yes? They are afferent cells, but they're actually bipolar cells. We went over this in the first section. So that means that they have like a cell body like this and one long process that goes out into the periphery that is gathering that information from my Ruffinian corpuscles and my Merkel's discs. And the other input, the other output, is going into the spinal cord. Is this the dorsal or the ventral horn of the spinal cord right here? Dorsal horn. Dorsal horn is important for what general modality? Sensation. And the ventral horn is important for what? Motor function. Okay, so the cells of the dorsal root ganglion are cells that bring, that bring afferent input from all sensory endings into the spinal cord. So their axons, their cell bodies are here, their dendrites are out here, bringing information in, their axons go up here and go up the somatosensory pathway. Yes?
What does the ventral horn do again?
Motor function, right? What are the cells that make up the ventral horn of the spinal cord? Alpha motor neurons, perfect. Must know this distinction about the spinal cord. I might even ask you to draw the spinal cord and label what is the dorsal and the ventral part. I would expect you to be able to draw this dorsal root ganglion, this kind of bulge with inputs coming in, and then these inputs, these outputs from the ventral horn. What is that bringing out? What is that doing? Yes?
It's coming from the brain and telling the muscles what to do.
Right. It's the motor output from these alpha motor neurons that you just told me about. It's the axons from these alpha motor neurons whose cell bodies are here making this gray part here instead of white. White is white axons. It comes out, they form the ventral root of the spinal nerve, and they come together with the sensory part. So the whole spinal nerve is made up of both sensory and motor parts, right? If you go in a little bit closer, this is all motor, and that is all sensory. But when it comes together, here is the full spinal nerve. It includes both sensory and motor parts. Is that clear? Anybody have questions about that? You're going to need to know that anatomy of the spinal cord with respect to both the sensory and motor aspects of the spinal cord. Okay? Yes? The ventral is efferent, right? It's sending input, sending information out, and the dorsal is afferent, bringing sensory information in. That's correct. Yes. Did you have a question?
Can we go over transduction
Inside the motor system? So there is no transduction.
So it just goes from the brain.
Yes. So motor function, so we're going to get into that. So that's a perfect question, because the motor system, sensory system is all about bringing energy information from the outside in, right? Light is coming in, force is hitting our body, and sound is hitting our ears. Motor is about the output, about moving our body around, and so it's a whole different modality. I mean, the idea is that sensory input comes in so we can understand the world and move accordingly. So, you know, lion is coming at us, we run and save ourselves. Okay, so, but you raise a good point, because we did go over an input side of the motor system. Do you remember what that was? Kind of a sensory part of the motor system. Yes? The muscle spindles. What is that general pattern called, that function called of sensing your own movements? Feedback, that's one way to call it, but there was a specific word, yes? Proprioception, very good. So, proprioception. is the collection of information about body movements and position. So if you read that first little anecdote in the motor chapter of your book, we talked about a guy that had damage to all of his proprioceptive input fibers. So this wasn't the feeling fibers. This was just information about what the muscles are doing. And what happened was he couldn't walk anymore. And the story, the moral of that story is that it's not about only the output, but we are actually using our proprioception, our feedback, to be able to move in the elegant and intricate ways that we move. And we talked about two major kinds of proprioceptive receptors. You need to know this term, proprioceptive. Another definition is feedback. But proprioception is this afferent information about muscle movement. And we talked about two ways that we learn about this. One is muscle spindles. If you ask what are muscle spindles important for, it's sensitivity and detection of stretch. Stretch of the muscles. It could be just passive stretch or more active stretch, but that's what stimulates the muscle spindles. Lots of activity in your muscle spindles because they're being stretched all the time, and that's what tells the body about where we are in space and what we're doing with our bodies. The Golgi tendon organs are a second form of proprioception, and these are more sensitive to muscle tension. So it's not just a passive stretch. You actually have to be really holding some weight. Why? Because these Golgi tendon organs are on the tendons. Where does the tendons connect what to what? Muscle to bone, perfect. So if you stretch that, if you activate that, you really have more muscle tension. And this is more of a really safety mechanism. You don't want your muscles popping off of your bone. And that is what the Golgi tendon organ is for. But let me focus on the muscle spindles because that's what we use the most. Okay? So here is just the anatomy. Here is the tendon connecting the bone to the muscle, as you correctly identified a moment ago. And here is the innervation of that Golgi tendon organ sensitive to tension in this area. Here is the muscle itself. And within the muscle, we have spindles that make up the muscle spindle system. These spindles have a special name. They're called intrafusal fibers, and that contrasts to the extrafusal fibers that make up the main part of the muscles. Oh, here's another important question for you. Muscles that we're talking about, controlled by primary motor cortex, are a particular type of muscle. What type of muscle is that? Striated, great, perfect. It's also called voluntary. You need to know both of these terms. Voluntary and striated muscles are what's being controlled by primary motor cortex. Can you give me an example of another type of muscle that is not controlled by primary motor cortex? Smooth muscle. What is a kind of smooth muscle? The heart, okay? So our motor cortex does not control our heart beating. That is controlled by autonomic centers in the brainstem, and we are focused, and we don't need to know anything about that in the test, but you do need to know that these muscles that we're focusing on are striated voluntary muscles. Did you have a question?
Yeah, what are the skeletal muscles?
What are the skeletal muscles? Anybody? Striated. All the skeletal, that's a good question. All the skeletal muscles that are moving our biceps, triceps, quadriceps, hamstrings, those are all striated muscles. Why? Because I can move them. I can decide I'm going to move my little toe. I'm going to move my right hand. Okay. Another simple one for you. Biceps and triceps, are they synergistic or antagonistic? Antagonistic. What is an example of a synergistic? What is a definition of synergistic muscle types? They act together. Perfect. Was there a question over there?
Yeah, so just to clarify, the entire kind of purple part that's in the middle, it comes up.
This? this is a muscle spindle, and this muscular part is called an intrafusal fiber. Okay?
The blue part that comes and wraps around it, that's just a...
That is a primary afferent fiber that innervates this muscle spindle. So this whole thing is called a muscle spindle, and this is the stuff that innervates it. Okay? And another word, a name for this muscle spindle apparatus, this muscular part, is called an intrafusal muscle fiber. Okay? Okay, so now, but you ask a great question because we're going to get to all the stuff that innervates. This is a sensory organ, right? We're talking about proprioception that's helping us figure out where we're moving in space. So here is a muscle spindle, and we have two types of afferent input that's figuring out what state this muscle spindle is in. There is a primary afferent fiber and a secondary ending. The primary afferent fiber goes around the center, the kind of fat part of this muscle spindle or intrafusal muscle fiber, and the secondary ending goes on the end. And the only distinction, or the distinction that you need to know, is that the primary afferent fiber is sensitive to dynamic stretch. While the stretch is first happening, as it's getting longer and longer and longer, your primary afferent fiber is saying, okay, it's starting to go now. You're stretching, you're stretching, you're stretching. But if you stay stretched for a long time, you're still stretched. That's where the secondary endings come in, and they tell you about chronic stretch of your muscle. Okay? How do you get stretch from the muscle? The nice illustration in the book is, imagine your arm just resting here very nicely, and you have a big, heavy BlackBerry that gets put into your hand, and it goes down. And that is stretching my bicep muscle right here. And right now, I have my, well, right now, as I'm moving, it's getting stretched, I have my primary afferent fibers saying, you're stretching actively right now. And then when I stay stretched right here, I have my secondary sensory endings telling me, you've reached a plateau, but you're still stretched. Okay? The last part of this is a sensory, a propriosensory apparatus that's telling you how stretched your muscles are. But let's say your muscles have to change all the time. I might passively stretch here, but then I move it up because I have to, bring something closer to my face. And then my bicep is contracted like this. And you can imagine that these muscle spindles have to stay sensitive to the state of stretch of my biceps. So they're very, very stretched here, and they're very, very contracted here. And the way that this intrafusal muscle fiber stays sensitive to that is because it has a gamma motor fiber here that is kind of keeping this, this muscle spindle at the same length as the overall muscle. So no matter what length the muscle is, you can always tell how much relative stretch you have. Because when you contract like this, the gamma motor fiber will go out and tell you, you're contracted, so get skinny. Not get skinny, but get short. And that's shown right here, where a muscle, this is just normal length. Muscle is stretched, you get lots of activity in your muscle spindles from your primary and secondary afferent endings. And then during the muscle contraction, here's a spindle that is very loose. And at this point is when the gamma motor neurons will go out and have this contract, so that this spindle isn't all loose and unsensitive, but it becomes now sensitive to the new length of the muscle. So think about that. Every time I'm doing this, my muscle spindles are changing to try and keep up with what I'm doing. And there are enormous numbers of gamma motor fibers just to help with this proprioceptive part. Any questions about that? Is that clear? Yes?
So the Golgi tendon organ is sensitive to both stretching and contraction? Because I was kind of...
Yeah.
I thought it was only sensitive to contraction, but it says like both receptors excited.
Yeah, it's how I would think about the Golgi tendon organ is that the Golgi tendon organ is really only stimulated when you have really strong tension on the muscle. It's not just like this. This isn't a lot of tension. But if you have me lift up a 25-pound weight and try and go like that, then I get tension on my tendon, and that's when the Golgi tendon organ is activated. It's not nearly as complicated as the spindle, and it's just you get a lot of tension, you get activation of the Golgi tendon organ to give you safety so that you don't pop your tendon off of your bone. Yes?
Might not be relevant, but I'm just curious, are those Golgi tendon organ and
Muscle spindles?
connections, are they, do they function on connections or on, how is that information transmitted?
Okay, that's a good question. It is, yeah, it's sensitive to stretch. So I don't know the answer to that question. I'm not sure if they know exactly how, but what you can show is that stretching will cause afferent information to flow through the primary and secondary spindle things. But good question, but you absolutely don't need to know that because I don't know the answer to that. Yes, in the back. The gamma motor neurons, no, I wouldn't call gamma motor neurons safety. The gamma motor neurons act to make the spindles maximally sensitive, right? So if the muscles are really long, you need to have long muscle spindles to match the muscles so that when it stretches from that point, you can then detect that. But then let's say you get to a really short point, and your spindles are really long, then they're kind of hanging out and not being able to be sensitive at all. So the gamma motor neurons provide a sensitivity valve for the muscle spindles. Does that make sense? Okay, great. Any other questions about muscle spindles, proprioception, or Golgi tendon organs? Okay, good. So that is the level of understanding that I want you to have. I would encourage you to focus on the muscle spindle apparatus and how that works. Okay, talked about pathways. Again, just like in the sensory system, the motor system has this particular pathway. It crosses, just like most of the sensory systems in kind of a sensory way, but we're going this way. We're going the other direction, from primary motor cortex down through the pyramids of the medulla, which is basically just a bump on the medulla, and then it crosses at the lower medulla to go down to the particular level of the spinal cord. And what does this neuron, this axon from primary motor cortex do when it gets to a particular level of its level of the spinal cord? Anybody know what it does? Yes? Sorry? I can't hear you. No, it's changing sides up here. So it's changing sides, it's going down. And so what it does is, what is the final common pathway for motor information to be conveyed to the muscles? What? Alpha motor neurons, right? Final common pathway, alpha motor neurons. That means that those primary sensory motor neurons have to synapse on the alpha motor neurons. So these axons from primary motor cortex are going down, they're crossing, and what they do is they synapse on the alpha motor neurons. And then the alpha motor neurons go out and what do? What do alpha motor neurons do? Muscle what? Muscle contractions. Is that what you said?
I said muscle junction.
Muscle junctions. Well, they form neuromuscular junctions. That's right. So what is a neuromuscular junction? Yes, when the axon of the primary alpha motor neuron meets a muscle fiber. What is the neurotransmitter used there? Acetylcholine, perfect. And so what these alpha motor neurons are doing are they're synapsing, making a neuromuscular junction, but the result of that, and acetylcholine does what to a muscle? Contracts it, perfect. Okay. Here is something that you also need to understand that we talked about in multiple different settings, which is the tuning curve. I know you talked about this a lot in your review sessions and with your TAs. We talked about sensory tuning curves. So can you give me an example of a visual sensory? What is a visual sensory tuning curve? Don't look at this, but what is a visual sensory tuning curve? Anybody? Yes?
It would show to what specific orientation the bar and neuron responds, or there's also like the bars or...
Right, perfect. It shows to what particular sensory orientation a particular neuron responds. It is measuring what this neuron pays attention to, what it likes to fire to, okay? And that's the basic concept of these tuning curves. You could have an auditory tuning curve. So what would an auditory tuning curve look like? You would be recording from a cell in primary auditory cortex. What would you test it on? Primary auditory cortex cells, yes. Different frequencies, perfect. If you're looking at spatial localization, you can test it on sounds in different locations too, yes.
So the significance of this basically is that a single neuron doesn't respond all or nothing to just one frequency, but it has a curved line response to a variety of, I guess in this case, movements. So
Well, yes.
It responds to 180, but it also responds to 90 less so.
Yes. The main point is for you to understand how we measure the different features of cells in the brain. Sensory tuning curves. If you recorded a visual cell and played it sounds, what would you have? What would you see? Nothing. You would get no sensitivity at all. And if you showed a cell in primary visual cortex faces, what would you see? Kind of weird stuff. But if you show it lines and orientations, then you would get these huge movements. That cell is sensitive to that kind of thing. And you're right, what we're showing is that it's not all or none in this case, but some cells are very, very sensitive so that their tuning curve would go like this, woo, and come back down. This one has a broader tuning curve. But the general concept is that we're trying to get you to understand that different parts of the brain have sensitivities to different things. If you record auditory information in the visual cortex, you don't get anything. If you record the wrong kind of visual information in the visual cortex, you don't get very much. But if you record the right kind of thing, you get a very specific tuning to that particular feature. Okay? So you should understand what a visual tuning curve is, what a auditory tuning curve is, and this example, what a motor directional tuning curve is for primary motor cortex. The other important concept that I do want you to understand is plasticity. And it's important to understand plasticity in the sensory system because we're going to really focus on it a lot when we get into our cognitive learning and memory systems. So we went over multiple different aspects of plasticity. Who can give me a definition of plasticity or even an example of plasticity that we talked about? Yes?
They would cut off certain fingers of the monkeys they tested on, and then their brain would change its neuron connections to make up for that.
Perfect. So she said an example was when I told you about they would cut off a finger of a animal and then record in primary somatosensory cortex, and you would see these neurons start to have very different patterns of activity. So plasticity is changing of a neural representation, changing of activity in the brain as a function of changes in the environment. So yeah, removing your finger is very different. Here's another example of plasticity that we talked about in the motor system, where here is the green. This is a slab of a rat motor cortex. And the green represents the electrode areas where they got responses to either wrist movements or little rat finger movements. Paw, I don't know what you call little fingers on a rat paw, but those movements. Then they trained the animal to do a very, very intricate discrimination where he had to go in, turn his wrist, and use his little rat fingers to get a reward. He got really, really good at that. And then you go back and record and look at all the huge area that is now representing the little rat wrist and his little fingers. That is plasticity. The brain has changed as a function of this learning of a motor function. Yes? What's the difference between green and blue? Yeah, the green, so you see the blue and the green, they have these little circles on it. All those little circles are individual electrodes. So they're measuring activity in these different parts of the cortex. So this shows a chunk of motor cortex in the rat. And the blue represents all the areas that were recorded from. And the green, in particular, is just the areas that are representing the wrist and the fingers. Okay? So you can record, and you record here, and you say, okay, the rat's moving his wrists and fingers. Do I get activity? Yes. Move here. Do I get activity? No. No, no, no. And then only the green, you got activity with this particular kind of movement of a particular area. So let's say the rat is moving his right paw. Where are these recordings? Left primary motor cortex. Good. And so that's what you see in the left primary motor cortex. Then you give him his training, the little rat his training, and then you record again in these same sites. And again, the blue is where you record, and the green is where you actually got these this activity for wrist and hands, and you see how much it changed. That is an example of plasticity. Just to cycle through, here is the example that you pointed out about what happens when a digit is amputated. You change something in the environment. This is pretty drastic change. And what you see is where digit 3 used to be represented on the cortex, you see an expansion of digit 4 and digit 2. So digit 3 disappears. There's no more representation. And in that spot that used to be digit 3, digit 4 and digit 2 move into that spot. Another example of plasticity. What are the fingers are fused together? What happened there? Anybody remember? So you can actually super glue the fingers together and have them do everything with two fingers together. Do you remember what happens to the representation in their cortex? Yes? Perfect. The areas of the cortex merged as if it was one digit, and that's shown here. So here is another very nice example of plasticity. Lots of things are happening in your cortex. The things that you're doing every day. You start texting a lot, you're going to have big representations of your thumbs, both in your primary motor and your somatosensory cortex. You start playing a new instrument. It changes the patterns of firing and the sensitivity of your motor and sensory cortices. This is happening all the time. And finally, a last example is of plasticity this is from the auditory cortex. We went over this in class, where before training, a particular cell in primary auditory cortex had sensitivity to a particular frequency of sound, but then the animal got trained to discriminate a particular sound to get reward. And this was a reward he really liked. He got really, really good at it. And look what happened to this sound, this cell. This cell seems to have gotten recruited to now respond best to the learned frequency. This change happened as a function of learning. Another example of plasticity. Does everybody understand how this experiment was done? Anybody want another explanation? Yes?
Not so much of how it was done, but the individual cells
Yeah.
change what they respond to, and the cells
Well, that's a good question. So these experiments were done using a particular tool. So the question was, she said, well, you just told us that the representation of these cells changed, but their anatomy doesn't change. What's changing? And the answer to that is that the tool used on these experiments and these experiments and these experiments is physiology. We are looking at the firing rate and action potentials of a single cell and simply measuring what response it responds highest to. Can I go in and then fill the cell and see what it looks like now? We could, but then we didn't know what happened, what it looked like before. So we don't know the answer to that. There is there are techniques that allow us to visualize changes, and we suspect that a lot of these changes are underlined by changes in synapse formation, changes in the size and shape of the dendritic or axonal arbors. Those are much more difficult questions to ask. But yes, the answer is it is underlined by a physical morphological change. But the easiest way we have to measure this is by just the response, physiological response that the cell is giving. That's a really great question. Any other questions about this? Okay, so just a couple of things to jump to the second of our two motor lectures, talking about strategies to reconnect a damaged spinal cord. A huge problem, a huge health risk, particularly for young people. And we talked about multiple different ways. They are looking at strategies. None of these work perfectly, and these are still experimental. Providing stem cells to differentiate into new neurons. Does everybody know what a stem cell is? Who can tell me what is a stem cell? Yes? A cell, say that first part again? Right, a cell that hasn't yet determined its specific function, but even more importantly, it's a cell that can potentially differentiate into any other possible cell, into a cell that can have the connections to to replace the neurons that are damaged and replace the axons that are damaged, and then go and make the right connections. Okay, so that is, and anybody know where stem cells come from? Yes. Bone marrow. The most common and controversial place that stem cells come from are fetuses. So all fetuses have stem cells. They start from stem cells, and those stem cells differentiate into every possible cell of our body. They can differentiate into skin cells and primary motor cortex cells. And all we need to do, small task, is figure out the molecular and the chemical signals to tell it to turn into what we want, and then to connect in the way that we want. And that is why they're so exciting to scientists, because they could cure so many different diseases if we were able to do that. But they're also very controversial. Anybody know why they're so controversial? Yes?
Because it damages the fetus, and people who believe that life starts at conception think that's sort of like a form of murder.
Right. Well, I mean, we don't kill fetuses just to get stem cells, but we use them from aborted fetuses. So that's a very, very controversial and charged subject. And you're absolutely right. People have a big problem with using these fetuses for anything. And so that's why it's so controversial. Now, there are stem cells in you and I. We have stem cells. We're going to talk about how there are two parts of the brain where new neurons, new brain cells are born, even in adulthood. Anybody know what those brain areas are? There are two brain areas. One of them is the hippocampus that we're going to be able to talk about for about 2 minutes today. And important for long-term memory. And the second part of the brain where new neurons are born is called the olfactory bulb. Okay? And so you and I are both birthing new hippocampal and new olfactory cells. all the time. And so potentially we might be able to use those cells as stem cells, but I don't want somebody digging around in my brain to get my stem cells to do that. And so again, the fetal stem cells become very, very valuable for lots of different reasons. Their ease, their availability, and their potential. Our adult stem cells that differentiate into new cells, they can only differentiate into two kinds of cells, hippocampal cells or olfactory cells. We might be able to figure out a way to get them to differentiate into other cells, but those are some of the interesting questions that people are dealing with in, as we address questions of spinal cord injury. Okay, second kind of way that we can address spinal cord injury is transplantation of glial cells. Glial cells are these helper cells that can help provide a medium over which new axons can grow, a medium that can grow over the damaged part of a spinal cord, right here, shown in gray, darker gray right here. And they're exploring that, again, still experimental. Neurotrophic factors. There are very important chemicals that we know that exist in the brain that promote growth and kind of attract new axons to come in a particular direction. And if we can use those in the right way, we might be able to promote axonal growth across the damaged region. Still don't know exactly how to do that, but that's being explored. And sorry, I keep going the wrong way. And then finally, there is the idea of transplanting peripheral nerves across the damaged area that we talked about. You should know the difference between polio and ALS. They both damage motor neurons. And the last thing that I'll focus on before we move to just 15 minutes of learning and memory is diseases of the basal ganglia. You need to know the parts of the basal ganglia. Use your atlases. Oh, sorry, yes?
What is the difference between polio and Lou Gehrig's? Like, both of them result in death of cells in the brain and spinal cord, right?
Yeah, so polio is a virus that destroys motor neurons of the spinal cord and brainstem. And basically, ALS is even more death-inducing to even more kinds of motor neurons, including primary motor cortex, neurons in the spinal cord. So they're both killing motor neurons, but I would say ALS is even more deadly and causes what's called shut-in syndrome, where your cognitive functions of your brain are just fine, but because you have no motor ability to talk or to move anything, you can't even communicate. And so it's a terrible, terrible disease. And again, I'll recommend a beautiful piece written by Tony Judt, who is suffering from Lou Gehrig's disease, called Night, to give you a feeling for what that disease does to people. Any other questions before I focus on the basal ganglia? Yes?
For the strategies to reconnect the spinal cord,
Yeah.
have any of them been proven to work, or are they just all being researched still?
Well, I mean, there's a lot of work with stem cells. Do they absolutely work? There's some progress. So there is some in animal models. There is progress in being able to get the rats to have a little bit more motor function. Can they get up and run around like other rats? No. And do we have a way to translate that to humans? No. But that's where we are in the research. And I know there's lots of just rehabilitation centers that actually use not so much these kinds of strategies, but motor exercise strategies to just try and get both the muscle movement going, but also try and engage through just physical therapy the parts of the brain that might be able to take over for the parts of the brain and spinal cord that might be able to take over for the parts that were damaged. And it is actually amazing that people initially that can't move at all, that seem completely paraplegic in some cases, can really regain a large amount of their motor functions. And it remains unclear. You know, damage, it's not like everybody has the same clean damage. There's car accidents, there's, you know, falling accidents. You're doing all sorts of weird things. And depending on exactly how your accident works or what your accident did, you could have more or less potential for these kinds of physical therapy approaches to help you. Any other questions about that? Okay, so basal ganglia, caudate in a little tail going all the way around here, putamen more laterally, globus pallidus is this darker part, subthalamic nucleus, and substantia nigra. What is the major neurotransmitter used by the basal ganglia system? Yes? Dopamine, yes, very good. And where is dopamine that is projected, that's activating the caudate and putamen? Where is that produced? Substantia nigra, very good. So here is the diagram that I want you to know and understand, because it really gives you the full picture of Parkinson's disease. Here is a very simplified circuit of the basal ganglia, where you have large areas of cortex, including motor cortex, but many other forms of cortex that project to the striatum in an excitatory way. Striatum, globus pallidus to thalamus, inhibitory projections. Striatum, inhibitory to the globus pallidus, inhibitory to the ventral lateral thalamus. But into the thalamus comes in dopamine. It is excitatory to the striatum. And this substantia nigra, that is a structure that is damaged in Parkinson's disease. You need to know this. Striatum, the cells of the substantia nigra that produce dopamine, those are the cells that die in Parkinson's disease. So let's look at what happens. You get less dopamine, you get less excitation to the striatum, so the striatum can go and inhibit less. If you inhibit less, you excite more. What happens in Parkinson's disease? You can no longer control your motor movements. And that movement and that shaking that is so common in Parkinson's disease is thought to be due to a release of inhibition. You don't have enough inhibition because the poor striatum isn't getting its jolt of excitatory dopamine. Everybody get that? Okay? So we talked about it last time. You came up with fantastic ideas. What if you have a death of substantia nigra cells? What are we going to do to try and improve that, to try and change that? L-DOPA. You put more L-DOPA so the neurons that are left can make even more dopamine. Any other ideas? Smoke cocaine. That could make you feel better, maybe, about the... I think that is a brilliant suggestion, and using all the information that you got from our lectures on drugs in the brain. But cocaine is not approved to treat Parkinson's disease now. You could start a whole new movement here. There are other ways. Great idea. Love that idea. Creative, yes. Deep brain stimulation, and that's what works is deep brain stimulation. What can work is deep brain stimulation of the subthalamic nucleus, because that goes in and it doesn't stimulate the striatum directly, but it's also stimulating the globus pallidus. That's helping with this inhibition, and it's stimulating the substantia nigra even more. And so. The other idea that you guys came up with last lecture is you can implant stem cells into the substantia nigra, try and get them to differentiate into dopamine-producing cells, and there you've solved your problem. You also have to hope that those dopamine-producing cells then project to the right area and get the right infusion of dopamine to actually improve the symptoms. And that's what can happen, but there are lots of problems with and side effects from implanting fetal stem cells into the substantia nigra. There is epilepsy that can develop. And it's probably because these cells then differentiate into different things and do wacko things to your poor substantia nigra that makes it even worse. So it can really help your symptoms, and it can't. We don't know enough about it to control it 100% of the time, and that is what we're working on to try and understand in terms of the stem cell research that so many people are doing. Okay, right, okay, so any other questions? What I've gone over for you, particularly for the motor systems, is what you need to know. Focus on those spindles. Focus on understanding antagonistic versus synergistic. Focus on, you need to know the anatomy and the function of the spinal cord. That is key to our understanding of sensory and motor systems. What does the dorsal root ganglia do? Where are alpha motor neurons found? What pathways are going out? Are those pathways afferent or efferent? There are beautiful illustrations in the book. Just make sure you know the anatomy and where these key elements sit in the spinal cord. Any other questions? Again, I encourage you to talk to your TAs if you have questions. Email them. Email me. Come and see me. Email me for an appointment between now and Wednesday. I'm happy to talk to you, not just before the exam. I'm happy to talk to you anytime about questions that you have. And now for, let's see, 8 minutes, we will talk about learning and memory. Okay, so basics of learning and memory. Just some key definitions. Learning is the process of acquiring new information, and memory is the ability to store and retrieve information. The idea is that specific information that you know that alpha motor neurons are final common pathway to move a muscle, that information is stored someplace in your brain. Okay. History of how our understanding of memory. It goes back to something we talked about in the very first lecture. Anybody remember what phrenology is? Phrenology. Yes? Perfect, perfect. So it's where you'd go and measure the bumps on the head, because the idea was the bumps indicated how well developed the brain area right below those bumps were, and you would try and decide by the bumps on the head whether you were a brilliant person, whether you were a psychopath, whether you were a great musician, all these different things. But the underlying idea there was that you could tell that different parts of the brain had specific functions. It was called localization of function. So that whole era had kind of a sour taste to it, because of course you really couldn't tell anything from the bumps on the brain, so it kind of became like psychic reading, going to the phrenologist. But it was underlying an idea that we now know is true. That is, there is localization of function. But because the phrenologists were ridiculed, then we also threw the baby out with the bathwater and said, oh, well, those phrenologists were wrong. Not only were they incredibly wrong about lumps on the brain, but they're also wrong that different brain areas had different functions. So we went back to an idea that actually our brain function is so complicated, it can't be localized. It's kind of this big, complicated neural network. And they didn't use that word back then, but they were thinking that way. And so we really can't understand particular functions defined by different parts of the brain. And this was particularly true for higher cognitive functions, because during that time, people were starting to understand that, in fact, primary occipital cortex was important for vision. Didn't do anything else, get damage back there, you're blind. You still think, you can still hear, you can still speak, but you're blind. But that's a sensory kind of function. We're talking about memory here. That is more higher level. That is more cognitive, and I really don't think that that can be localized in the brain. And so that was kind of the area, that was the environment into which Carl Lashley, a famous professor from Harvard, started his studies in the 1930s. I love this picture of him. He looks like the prototypical Harvard professor. I think that was really those little pincer things that he took off. But he looks like a very learned guy, and actually was very, very smart. And he did a series of systematic studies that were wrong. So let me tell you what they were, and let me tell you why they were wrong. So he wanted to study the brain basis of memory. And this is a question that you guys could study right now. And so he taught the rats something. He taught the rats something and then tested their memory for it after damaging part of the brain to see whether that part was involved. Here's what they taught. The rat got put into a maze like this. This is just an overhead view. And the rats had to learn how to go this way and then go through this door and turn left and go this way, right, turn this way. And then the food was right there. And he measured how fast they were able to go through this maze. That's learning, right? Would you all agree that's a good way to test learning, how fast you can learn how to do this maze? You think that's a reasonable way to test learning and memory? Yeah. Okay, so he tested all the rats. And then what he did is he looked at brain lesions of different sizes. And he was focusing on the outer surface of the brain, the neocortex. Okay? And he made small lesions, and he made bigger and bigger lesions. And he looked at the errors in, so he taught the rats the maze. He gave them a lesion of different sizes. Small lesion, medium lesion, large lesion. He tried different parts of the brain. And then 20 days later, he retested them and looked at the errors during relearning after 20 days after surgery. Doesn't matter how long, but he just looked after surgery. What he found was this interesting correlation. And the correlation showed that the more brain area you removed, the more errors that you had. And all of these different points were different brain areas. Small lesions, no matter where they were, did not seem to result in any bigger impairment of memory for this maze. But what did seem to matter is you give them a big old lesion in the cortex, and then, okay, then they all started to have problems. What do you think that means? What would you conclude if you were him? That memory is localized or memory is not localized? What do you think? Not localized. What would you expect if it was really localized? Yes?
Sharp drop-off in their ability if you punched a hole in a certain place.
Perfect. A sharp, huge impairment if you put the lesion in one place but not in other places. Does everybody get that? Okay? So that's what he was testing. That is a perfectly reasonable question to test. And what he found was not what you just said, but what you just said. That is, there's not a localization of function. It's the bigger the lesion, the more impairment of memory that you have. And from those data, he developed a law called the law of mass action in the cortex for memory. And the law said that the severity of the memory impairment from maze performance correlated with the size of the cortical area removed and not with its specific location. Okay? Law of mass action. What this basically said is that there's no localization of memory. That memory seems to be this thing that's involving more and more cortex. No matter where you damage, you get a little bit of impairment. But if you damage enough of it, then you only then do you get a big impairment. And I don't have enough time to tell you why that was wrong, but next Wednesday I will tell you all about why that's wrong. Please come and see us, come talk to us, come email us. If you have questions for Wednesday, good luck, and I will see you here on Wednesday morning at 11.